Hi, good afternoon, everyone. Sorry for being late. I was running from the other session. We are coming towards the end of our congress, but we still have a very interesting session. And this particular one is actually a special lecture. I purposely choose this topic of my interest. I think it's actually very important to our current as well as future practice. So we are going to talk about genetic testing in individual with kidney diseases. And I managed to invite Professor Ali Gharavi. He's a professor of nephrology and hypertension, chief of division of nephrology, and director of Center of Precision and Genomics at Columbia University of the States. So I actually do not know him, but I actually emailed to him after reading his publication, and he replied me very fast to say yes. Of course, today, because of the work commitment, he will not be able with us today, but I will invite you to still post questions. If you have any question, I promise I will bring it to him, get him to answer that, and then I'll post it in the platform later on. So with that, we will listen what Prof. Dr. Ali Gharavi is going to tell us about genetic testing in individuals with kidney diseases. Professor Ali.
Hello, it's a pleasure to attend the Malaysian Society of Nephrology. I wanted to thank the organizers for inviting me to talk about genetic testing in kidney diseases. I'm Ali Gharavi from New York City, Columbia University. These are my disclosures. So we're talking about genetic testing today, and this is all possible because we have been able to produce drafts of the human genome. Actually, the first draft was produced back in 2001, and now we are celebrating the 20th anniversary of the first draft of the human genome. The human genome has about 3 billion nucleotides of letters in its alphabet, and it took 10 years and $3 billion to produce one draft of the human genome. But since that time, advances in genomic technologies has enabled us to do this much faster and at a much lower cost, such that now we're able to produce, sequence individual genomes in about 24 hours at the cost of about $1,000. So as a result, hundreds of thousands of people's genomes have been sequenced. And so we are able to now examine these individual genomes to try to identify the range of genetic variation that's present across human populations and discern the type of variation or typos in the genome that are common and probably benign, and those that are pretty rare and is potentially disease causing. And that is the real challenge nowadays in genomics, is to be able to discern and interpret genomic variation, to be able to detect pathogenic changes that is responsible for different human diseases. So if you start to think about applying genomics to nephrology, you have to think about what is the spectrum of diseases that we are dealing with. On the left, you have the prevalence of end-stage kidney failure among children in the United States. The common causes of kidney disease are congenital cystic diseases, or primary and secondary glomerular diseases in children. On the other hand, in adults, still, primary glomerular diseases and cystic and congenital disorders are pretty common. However, secondary causes such as diabetic and hypertensive nephropathy are the other leading causes as the population ages, as could be expected. Another important category among patients with kidney disease is CKD of unknown cause, which I will talk to you about. And these contribute for about 5 to 10% of cases, depending on populations studied. So what's the prevalence of monogenic kidney diseases? Well, so far we had been able to sort of estimate this based on clinical diagnosis of genetic disorders. And this is a review in The Lancet published by Olivier de Vust, estimating that based on the published literature to date, that about 10% of kidney diseases are due to monogenic causes. And this was also reproduced in a study looking at the Australian population, where again, these authors estimated about 10% of cases of CKD are due to monogenic kidney diseases. Nowadays, we're able to sequence individuals to try to get this estimate not just based on the clinical diagnosis, but also based on the sequencing data. So So what are the range of monogenic kidney diseases out there? Well, a thorough catalog has not been completely developed. We set out to do this, and we're looking at the literature and various genetic databases, and we were able to catalog at least 625 million kidney and genital urinary disorders. These are monogenic disorders, and these can be categorized into dominant or recessive diseases. Most of these genetic diseases are recessive diseases. Most of them are congenital and developmental disorders. And you could see that these are pretty heterogeneous, and there are many different categories of genetic diseases affecting both electrolyte, metabolic, glomerular, tubular, interstitial diseases. So with this set of genetic disorders in hand, now you can start to go back, and if you sequence the human genome to see if they're in patients with kidney disease, you could see if patients may have potential pathogenic mutations in these genes. So another way to think about also the architecture of genetic kidney diseases is to look at the contribution of variants that are very rare and have a very large effect. So this is a classic depiction of this paradigm, looking at the allele frequency of a particular genetic variance from very common to very rare and the effect size. So when you think about a genetic variant that has a very large effect size, typically contributing to a monogenic kidney diseases such as polycystic kidney disease or Alport syndrome, these mutations tend to be really rare in the population because they confer a very large impact on phenotype and are subject to purifying selection. In other words, they're not easily passed on from one generation to another. And if you ascertain different families with the same disease, typically they have different genetic changes in the same gene, indicating that this is not a common variant in the population. So what I'm going to talk to you about is this category of diseases, therefore, where there are really rare mutations with very large effect. And these are different monogenic diseases: cystic diseases, FSGS, Alport syndrome, congenital kidney diseases, or tubular interstitial diseases. And this is a list of some of the genes that contribute to some of these phenotypes. The overlapping circles indicate that since we've been sequencing patients, oftentimes we see overlap in the clinical presentation of the phenotypes. And as a result of this, then, these clinical categories of disease tend to be blurred now that we have genetic ascertainment. And I'll give you some examples as well. And of course, CKD of unknown cause can be attributable to mutations in any of these genes, so depicted here by the large gray circle. So I'll talk about this in a little bit more detail. So when you're trying to sequence genomes, you can try to think about sequencing the entire genome, or 3 billion letters of the genome. But the real sweet spot in genome interpretation, the area that we can interpret the best, are the coding portions of the 20,000 genes in our genome, which compose about 1% of the entire genome. So this area, the coding portion of the called also the exons, can be selectively captured and sequenced. And so this is referred to as the exome. So when you think about the exome now, which contains the 20,000 genes, the coding sections of the 20,000 genes. Of those 20,000 genes, about only 4,100 are securely associated with the Mendelian disorder. And sometimes these are referred to as a Mendeliome or as a medical exome. And so if you start to look at the Mendeliome and all these 4,100 genes, about 600 to 700, as I mentioned, are associated with a monogenic form of nephropathy or urogenital disorder. And so this is sometimes referred to as a nephrome, and there are ways of selectively sequencing the nephrome, or even subpanels that address genes that are associated, for example, with cystic diseases or with FSGS or nephrotic syndrome. So this gives you an idea about the range of sequencing. It can be very broad, or it can be very focused on a smaller number of genes. The sweet spot right now in genome sequencing and interpretation is the exome, because now you have a pretty comprehensive view of the genes in the genome, including potentially novel genes that could be discovered in the future if you start to sequence this and keep the data. So we started to look at the utility of exome sequencing at adults with chronic kidney disease, looking at two populations, one from Columbia University, recruited for a biobanking study, and another one in collaboration with AstraZeneca, the Aurora cohort, which has about 1,100 cases of patients with end-stage kidney disease. This was a clinical trial of statins and ESRD. We combined the data set, we performed exome sequencing and variant annotation was performed based on the American College of Medical Genetics diagnostic sequence interpretation. This is a set of guidelines that have been issued by the American College of Medical Genetics for sequence interpretation and detection of pathogenic variants. To the right, you see the clinical characteristics of the patients. 90% of them were adults. It was a pretty good male and female representation, pretty equal. This was a multi-ethnic cohort representative of the population that we serve in the United States. And there was a range of clinical diagnoses including cystic and congenital diseases, glomerulopathies, diabetic nephropathy, hypertensive nephropathy, tubulointerstitial diseases, and nephropathy of unknown cause. And of note, 30% of cases had a positive family history of kidney disease, which is standard for most studies of chronic kidney disease. So this is the bottom line. Altogether, we had a 9.3% diagnostic rate, and we identified 66 different genetic diagnoses in 307 patients. About 2/3 of the cases were attributable to mutations in PKD1 or PKD2, which are a diagnostic of polycystic kidney disease, dominant forms of polycystic kidney disease, and type 4 collagen mutations typically associated with Alport syndrome of thin basement membrane disease. 3% of the cases were due to mutations in UMOD associated with autosomal dominant tubulointerstitial disease. And then another 37% of the cases were attributable to mutations to 60 different genes, so essentially ends of ones across the entire population, showing that there's a lot of heterogeneity. In addition, the overall diagnostic rate was 9.3%, but the rate really varies if you start to stratify patients based on the clinical diagnosis. So the diagnostic rate was about 1 to 2% if you looked at patients who came with a clinical diagnosis of diabetic kidney disease or hypertensive nephropathy, but it was nearly 25% in patients who had a clinical diagnosis of cystic or congenital kidney disease. So it kind of makes sense, and he told us that the clinical diagnosis really matters. In addition, we had good diagnostic yields for tubulointerstitial diseases and glomerulopathies in general. But very surprisingly, and we were very pleased by that we had a 17% diagnostic rate in patients with kidney disease of unknown cause. So this result is not unique. There are many, many studies that have now been published, and this is summarized here in this review by one of my colleagues, Enrico Cocchi, which summarizes essentially most of the studies that have been published up to that point. So this is roughly last summer, showing the diagnostic yield based on the clinical category, but also based on the sequencing modality that was used, both in pediatric and adults. And roughly the diagnostic rate can be as high as 60 or 70%, depending on how you select the population. The more carefully selected the population, the higher the diagnostic yield. So let me give you a case study. This is a 57-year-old who was newly diagnosed with kidney disease that had proteinuria. He had a kidney biopsy which showed focal segmental glomerular sclerosis. He was advised to take a course of steroids, and he came for a second opinion. We reviewed the biopsy, which suggested that there are some secondary features on it. So instead of rebiopsying him or giving an empiric course of steroids, we wound up doing exome sequencing. And it turns out that he had a dominant form of Alport syndrome due to a type 4 collagen mutation. And so the treatment there was to avoid steroids, referred for a new clinical trial that was targeting patients with Alport syndrome, and then start screening family members who may be at risk because this was a dominant disorder, and also thinking about who may not be carrying a mutation and could be potentially a donor for this patient. So just to tell you that the classic presentation for Alport syndrome is hematuria, sensorineural hearing loss, and ocular changes. But as we're sequencing more people, we're realizing that this is probably not the most common presentation. Mutations in 3 different genes can cause Alport syndrome. The classic presentation is most commonly seen in collagen 4A5, which is on chromosome X, responsible for the X-linked form of disease, which affects male men predominantly. But now these mutations in the call4A3 and A4 are increasingly recognized as an important contributor. And so when we started looking at the phenotypes that are associated with pathogenic variants in these patients, we had a total of 92 such cases. Only 40% of cases or so had a clinical diagnosis of Alport syndrome with thin basement membrane disease, whereas the rest of the patients had a diagnosis of FSGS, of GN of unknown cause, of CKD of unknown cause. So really, again, showing that the most common presentation for these type 4 collagen mutations is not necessarily the classic presentation of Alport syndrome, but all these non-specific cases. And this is probably the most common genetic findings in patients with FSGS or the family history. And so such patients often come with a history of being exposed to steroids, and we wouldn't have expected them to respond because, of course, this is a structural defect in the glomerular basement membrane and would not be expected to respond to immunosuppression. So this is something that we could help our patients with by avoiding exposing them to toxic medication by doing this type of testing, and a real benefit to start with. This is another example. This is a 25-year-old who has a family history of kidney disease since childhood. Kidney biopsies at age 8 and 11 were non-diagnostic. So basically he has proteinuria and an unknown cause of CKD. And he had 2 brothers who were similarly affected. He was getting married, and he wanted to know what would be the risk of transmitting this disorder to his children. And so rather than doing, again, another kidney biopsy, we wound up doing exome sequencing. And it turns out that he has a mutation in CLCN5, which is a diagnostic of this rare disease, tubulopathy called Dent's disease. This is a chloride channel, which in the endosomal membrane on the proximal tubule, and mutations results in low molecular weight proteinuria, but also for reasons that are unknown to hypercalciuria, which then, as it progresses, leads to nephrocalcinosis and kidney failure. This is also associated with bone loss. So we wind up providing this diagnosis to the patient, and his 2 brothers also had the exact same genetic defect. And so this really offered them a clinical diagnosis after many years of uncertainty. The treatment here is a thiazide diuretic to reduce the hypercalciuria, high citrate diet to reduce the nephrocalcinosis. We referred them for a bone density study, but also this is an X-linked disease. And so we were able to, and females are very rarely affected. So we were able to tell him that basically he would not be transmitting this mutation to any of his male descendants, but his daughters would be obligate carriers. And so that's for the next generation, something needs to be considered for this. So this again showed the power of doing exome sequencing for a patient who had a kidney disease of unknown cause and who had gone to multiple biopsies with no clear diagnosis over the years. How common is this problem of CKD of unknown cause? This is a worldwide review looking at the burden of kidney diseases, across the world. There were somewhere around 700 million people that were cataloged as having kidney disease. And obviously, kidney disease due to diabetes, glomerulonephritis, or hypertension was pretty common. But depicted here in blue are the range of the patients who have kidney disease of unknown cause. Here, this is stratified by different age group. And on the y-axis, you see the amount of disability, which is not early death or the days of life lost to disability. And it turns out that these patients with CKD of unknown cause had the highest number of sort of morbidity and this disability adjusted life years lost. So really an important category for us to consider across all the practices in nephrology. And so we recently reviewed the diagnostic yield of exome sequencing for CKD of unknown cause. A total of 6 different exome sequencing studies had been published, and they reported a total of 443 patients. Overall, there was a 22% diagnostic yield for CKD of unknown cause. There were 47 different genetic diagnoses that were identified. You could see all these different genetic diagnoses in this the outside circle here, and these map to different clinical categories of disease, such as glomerulopathies, tuberointerstitial diseases, or congenital cystic diseases. But importantly, type 4 collagen mutations accounted for 1/3 of such cases. So again, really important to think about this class of mutations in our clinical practice. So One of the typical questions I'll get asked is, who do I refer for genetic testing? How do I think about genetic testing in the patients? Should I send everybody? So these are some of the telltale signs I think that would be very important. So you should think about the clinical diagnosis that you have. So in other words, if you have thought and evaluated the patient, then you could see what the diagnostic rate might be. For CACUT is about 20%. As I said, for diabetes, if you think the patient has diabetic kidney disease or hypertensive nephropathy, the genetic diagnostic rate is about 2%. And it's somewhere in between for other clinical diagnoses. So the kidney disease subtype is important. If there's a positive family history, the diagnostic rate goes much higher. So you should definitely ask for a family history of kidney disease. Young age of onset, particularly pediatric onset of disease, is also a very important an important predictor of positive diagnoses. And then the presence of extrarenal features. So if there's any evidence of systemic disease or multiple organs that are affected, then also increases your diagnostic yield. So these are the parameters that I think about when I try to think about who should go for genetic testing. And so the other question that I get is, should I send an exome if that's available, or should I send a panel for sequencing? Well, if you're able to, this is what we did with our study when we took the exomes and then created virtual panels and said, okay, what would have been the diagnostic yield if we compare these virtual panels to exome sequencing? Well, for most of the clinical diagnoses, such as congenital diseases, glomerulopathies, hypertensive or tubular interstitial nephropathy, a targeted panel that targets those set of genes performs almost as well as exome sequencing, not perfectly as well, but almost as well. As you can imagine, for nephropathy of unknown cause, exome sequencing performed the best because it encompasses all different genes contributing to different clinical categories of disease. I guess the other alternative nowadays is also to have just a very large panel of the nephrome panel, essentially, which encompasses many of the kidney genes across the different clinical diagnoses, and that might be another way to go for genetic testing. And such panels are increasingly available. So this is one way to start to think about genetic testing, which test to send. If you can't find an answer, you should not give up because new knowledge accumulates very fast. And so the data can be analyzed a year or two after you've received the sequence to see if there are potentially new genes that have been identified. There's new information about some of the variants in those genes that are available. This is an example of two studies. The original analysis of exome sequencing, the original analysis was done in 2012 and 2013, and the data were reanalyzed in 2017 to see if there's a gain in diagnostic yield. And so here, in this study, the diagnostic yield went from 24% to 46%. In this study, from 25% to 37%. And in each case, most of the increase in diagnostic gain was attributable to the discovery of new genetic diseases in the intervening period. And so as knowledge accumulates, then you're able to then reclassify a genetic variant, reclassify new genes as disease-causing and try to look for genetic mutations in these. Another important category is this clinical update and variant reclassification. There's new knowledge about pathogenicity of some variants. The variants can be upgraded and downgraded, and there are lots of recommendations that have come over the last few years about how to best interpret genetic data. And so as a result of this, up to 20% of variants can sometimes be reclassified in one direction or another, some pathogenic variants to non-pathogenic, and often some variants of unknown significance to pathogenic. And so that can help also in terms of increasing your diagnostic yield and changing your management. So you should be on the lookout for these changes as well, and can potentially request this from the diagnostic laboratories. So I'll close by just talking about one important aspect of genetic testing. So far, we've talked about genetic testing in the context of diagnostic testing. So a person has a disease, CKD in this case, and then you're trying to say, okay, here's a list of genes associated with kidney diseases. We're going to look to see if there's a genetic variant that's associated that can explain the clinical presentation. In this situation, if you find a genetic variant in a gene that's associated with a disease that you're suspecting based on clinical evaluation, let's say a genetic variant in an FSGS gene, a patient who has nephrotic syndrome, then this genetic diagnostics has a very high positive predictive value. On the other hand, contrast this to the situation where you do predictive testing, where somebody is otherwise healthy for some reason, they do exome sequencing, or because they want to do health prevention. And now you're trying to test whether they have a particular disease or may be prone to a particular disease. Here, you're testing a wide set of hypotheses about their potential genetic etiology of disease. And so in this situation, just like any other test out there, the genetic testing has a much lower diagnostic predictive value because there's a very low a priori risk of disease. So you have to keep this in mind. And so this is also illustrated by the study that we did, which we wind up looking at the exome of about 8,000 healthy controls, so self-declared healthy individuals who had exome sequencing as part of research studies. And we wind up looking at this exome to see if they have any genetic variants that may be diagnostic of a kidney disease in any of these kidney disease genes. So if we didn't do if we did some basic filtering as recommended by the American College of Medical Genetics, something around 22% of individuals had a set of genetic variants that may be diagnostic of a kidney disease. So that's a huge number of people, much higher actually than the prevalence of CKD in general in the population. So this was obviously a false positive finding. And most of this was attributable to genetic variants that were previously reported as being pathogenic and needed to be reclassified as benign. Most of these were probably too common in the population to be really plausibly associated with the monogenic kidney diseases. They were reported at that time, but we didn't have good genetic databases to look at for the frequency of genetic variants across different populations. And these need to be cleaned out of the databases to improve genetic diagnostics. However, even after we did very, very strict filtering based on allele frequency and manually curated the variants, we still were left with about 1% of individuals who had a genetic variants that were potentially pathogenic and potentially predict to be disease-causing. So now we didn't have, unfortunately, access to the health records for these patients to see if they actually had the corresponding clinical diagnosis. So a lot of work will have to be done to figure out whether these variants are pathogenic and the patient is not diagnosed yet or has the disease and needs to be really, clinically worked up to get the diagnosis, or maybe at risk of developing the disease somewhere down the line in the future, so they need to get follow-up, or whether this was also erroneously reported. So before we wind up doing exome sequencing and genetic testing in a predictive model, we'll have to come up with a much more robust analytic framework and a set of genes that we can better consider for this type of work. So I would say in summary, genetic kidney diseases are very common and can be present in about 10% of the kidney disease patients. The prevalence of genetic diseases varies by age, by family history, clinical diagnosis, and the referral population that was studied. And so the detection of genetic disease has many implications for disease management, including therapy, as I mentioned. And there's a lot of also additional useful information in genetic testing, such as secondary findings, rescue allele screening, and pharmacogenetics, which I didn't have time to go into, but you can read up about. And then for predictive testing, we'll have to wait until we have a better curation of variant databases, and we'll have to restrict this to a set of well-selected, well-studied genes. So if you want further reading, these are some references for you to follow up on. And I wanted to thank all of our organizers and collaborators for this as well. A lot of people have contributed to this at Columbia through this work, and I'm really grateful to all of their contribution. And I also want to thank all of our funders. Thank you very much.
Yeah. So thanks, Prof. Ali Gharavi, for the very enlightening lecture. I think the main message from this lecture is genetic kidney disease might be present in up to 10% of our patients with chronic kidney disease. And the diagnosing this kind of genetic disease might be important because it can avoid unnecessary investigation as well as management. For example, unnecessary steroids treatment in those patients who have presented with like FSGS. And I think this is something that we have to consider in our future management of kidney disease. And many of them might be having collagen disease. I do see some question coming up in the question panel. As promised, I will submit this to Professor Ali Gharavi and get him to respond to this. And I will upload it in the platform later on. Yeah, so with that, I would like to conclude the session by thanking everyone to join the session. So please go ahead to actually join our debates as well as a viewpoint session later. Thank you very much.